My name is Veleba, and I would like to present you my thesis about genome size and carnivory in plants. My supervisor is, as you know, the Associate Professor Petr Bureš, and as model group, we have chosen carnivorous plants. It's an ecophysiologically defined group of plants which have adaptations to kill and sort of digest other organisms and acquire nutrients and use them in their growth. The carnivorous plants evolved in several clades. I will shortly introduce them. The first one is Inidioderpoales, where in family Bromeliaceae and Eriocaulaceae there are a few species of carnivorous bromeliads and Pepalanthus bromelioides. Another clade is in order Oxalidales, family Cephalotaceae, which is monotypic, containing only Cephalotus follicularis, which is quite unique and distant clade of carnivory. While in order Ericales, there are two clades, family Sarraceniaceae and Rhodulaceae. Possibly the richest clade for carnivorous plants is the order Lamiales. It might be due to the abundant defensive glandular hairs that are present on many plants in this order and which serve as sort of a pre-adaptation for carnivory. We currently recognize three clades of carnivorous plants, family Lentibulariaceae, family Biblidaceae, and recently described genus, unfortunately without a picture, Phyllocoxia from the family Plantaginaceae. The oldest and most diversified clade of carnivorous plants lies in Caryophyllales. Apart from fully carnivorous clades or families like Doseraceae, Rhizophyllaceae, and Nepenthaceae, it contains also two non-carnivorous families, Ancistrocladaceae and Dionchophyllaceae, who evolved from carnivorous ancestor, but they lost their carnivory, with the exception of the strange Triphyophilum peltatum, which is sort of part-time carnivorous. Carnivorous plants share quite similar habitats all over the world. This is a peatland here in Czech Republic. This is an alpine vegetation in the Andes on the Peru-Ecuador border, where carnivorous plants grow as well. And this is an open highland forest in Sumatra, where also some carnivorous plants from the genus Nepenthes grow. So why they share these habitats? It's because of their traps, their main adaptation. Because traps, they bring benefits. They bring nutrients, and these nutrients can be used to enhance the growth, to increase photosynthetic rate, or to produce more seed that may enhance the plant's fitness. On the other hand, the traps are not optimized to photosynthesize effectively, and their construction and maintenance are quite costly as well. For these reasons, carnivorous plants are limited to certain habitats where nutrients are deficient, but there is abundance of light and water. Only there they can perform photosynthesis effectively enough to really use the nutrients they acquire through traps. I think there is One historical note, which should be mentioned here to clarify the relationship of carnivorous plants and genome size research. It was since about 1950s when genome sizes started to be measured, and for many years, Arabidopsis thaliana was considered as the species with the smallest genome among flowering plants. But then came the paper from Professor Greilhuber and his colleagues, and they analyzed some species from the family Lentibulariaceae and found that Genlisea aurea has genome 2.5 times smaller than the previously smallest genome in Arabidopsis thaliana. The genome size refers to the DNA amount in the cell nucleus. It's quite variable. In flowering plants, the largest genome is about 2,400 times larger than the smallest genome in Genlisea. When we talk about the DNA, the genome, we usually consider it as a code. It can be adaptive, it can be selected for best combinations, but it's not necessarily the only way the genome can affect the plant's evolution. The plain genome size, the size of the so-called book where the code is written, can be adaptive as well. Annual species can serve as an example because the duplication of larger genome takes longer time, and species which are limited by time has to keep their genome small in order to perform all necessary cell divisions in their life cycle. Similarly, species growing in nutrient-poor habitats, like carnivorous plants, for example, are supposed to be forced to keep their genomes small to spare nutrients, especially nitrogen and phosphorus, which are important building elements of the DNA. To analyze the genomes of carnivorous plants, we sampled cultivated plants from collections of plant enthusiasts and botanical gardens. We analyzed them on flow cytometers for the total genome size and AT-GC ratio of the genomic basis. We employed molecular phylogenies and constructed phylogeny trees because we performed statistics in a phylogeny-informed way. We also employed one other method, which was flow cytometric detection of holokinetism, but I will say a few details about this method later. Well, since the study of Professor Greilhuber and his colleagues presented only a small part of the species of Lentibulariaceae, we have tried to analyze this family quite extensively. The family Lentibulariaceae consists of 3 genera. The genus Pinguicula is quite remarkable only by the leaves equipped with glandular trichomes, which makes them an adhesive traps, but their body plan is standard. On the other hand, the genera Genlisea and Utricularia are quite unique as their severely altered morphology because they completely lack roots. The root-like structures in Genlisea are actually helically twisted underground leaves, which allow them to catch small aquatic or underground animals or organisms. And in Utricularia, the leaves are transformed into complex bladders which actively form vacuum inside, and upon stimulation they suck in small organisms as a prey. In this family, we have been asking what's the variation of genome size, and of course we have been curious if we could find some more miniature genomes like Genlisa aurea. We have been also curious how did the genome size and the genomic GC content evolved across the phylogeny of the family, or whether it is sort of co-evolved. The results are quite interesting. Even the largest genomes in the family Lentibulariaceae are quite small in the context of other angiosperms. As for the individual genera, the genus Pinguicula tends to have larger, while genus Utricularia smaller genomes. But the most interesting genus is Genlisa because with only 12 analyzed species, this genus covered the whole variation of the genome size in the family, the smallest and the largest plant in our dataset. We have found several species smaller than Arabidopsis thaliana, but unfortunately we haven't found any species smaller than the previously published Genlisa aurea. When we compare genomes of Lentibulariaceae with their sister clades or some other clades in Lamiales, we can see that Lentibulariaceae are quite clearly smaller. What's the reason? It could be either the nutrient limitation, which causes the genomes to be kept small, or it can be the mutation in COX enzyme, which has been described in some species of this family. This COX mutation is supposed to accelerate the ATP production for the traps, but it has the disadvantage of increased production of reactive oxygen. The reactive oxygen, therefore, can damage the DNA, and the repairs through homologous recombination combined with increased rate of deletions causes that the genomes of Lentibulariaceae can be in a state of complex and not truly complex, but constant shrinking. How did these genomes evolve? Well, in Pinguicula, we have detected a significant tendency in many species for genome growth, while in Utricularia, the genomes are constantly shrinking. It's again the genus Genlisa where the most interesting things happen. There is one clade with severe miniaturizations and other two clades where genomes are growing. Comparing the genome size and the GC content shows us that there is a clear positive correlation between these two parameters, but also the variation of the GC content is quite interesting because it's almost 11% and it covers the majority of the GC content variation in all flowering plants. This is probably the largest GC content variability in a single family. To conclude this, we can say that Lentibulariaceae have small genomes and miniature genomes are quite frequent. The genomic GC content is notably variable. What's behind this? It's not sure whether the genome miniaturization is a passive consequence of the COX enzyme mutation, or it might be an adaptive evolution due to nutrient limitation. If the evolution of the genome size is adaptive to nutrient limitation, we could expect a similar trend also in other carnivorous clades. So We analyzed genomes of carnivorous plants in the order Caryophyllales, with special emphasis to genomes of Droseraceae. The Droseraceae, which is one of the major clades of carnivorous Caryophyllales, share in the meaning of traps the ability of movement. In genus Drosera, the traps can move only slowly and mostly in limited way, but the movement in snap traps of Dionea and Aldrovanda is remarkably unique and very popular. Again, in this family, we have been curious what's the variation of genome size in Droseraceae, and quite expectedly, we have been curious whether we can find any genomes as miniature as in the previously researched Lentiboraceae. We have also wanted to compare the genomes of carnivorous Caryophyllales with their non-carnivorous relatives, and we wanted to model the evolution of the genomic parameters across the Droseraceae phylogeny. Last of our questions was whether any of the genomic parameters is adaptive and correlated to climatic variables, which were available for this family. This you have already seen, the family Lentibulariaceae, and this is Droseraceae. As you can see, the genomes of Droseraceae can be quite large, but even small, but far from being miniature as in Lentibulariaceae. When we add some other species from carnivorous Caryophyllales, we can compare them with other non-carnivorous Caryophyllales. You can see that the difference is visibly not very large, and indeed it's not significant. The genome size evolution in Droseraceae is not really exciting. There is one clade which tends to increase their genomes, while in the other the genomes are decreasing. But the truly interesting things happen in the evolution of the GC content. We have detected several clades where GC content is quite high and growing, and also other clades where GC content is low and still decreasing. When we compare the GC content with genome size, these parameters are obviously positively correlated. But again, the interesting thing is the GC content compared to the already mentioned climatic variables. In this case, the strongest correlation was with the temperature annual range, which is a difference between the highest and the lowest temperature in the distribution area of the given species. What does it mean? Well, it means that we have species like Drosera rotundifolia from temperate zone, which endures high fluctuations, high temperatures in summer and low freezing temperatures in winter. And these plants have high GC content. On the other hand, species like this Drosera schisandra growing in rainforest floor in Australia, where annual fluctuations are almost negligible, have GC content much lower. And what mechanism is behind this? It's called the stabilizing effect of guanine and cytosine, because guanine and cytosine in DNA is connected by triple bond, in the contrast to thymine and adenine, which are connected only by double bond. It has been proved that DNA richer in the GC, guanine and cytosine, has higher thermal stability, but in plants it seems that higher GC content seems to be connected with overall stability of the genome in fluctuating environmental conditions. To conclude the family Droseraceae, genomes can be considered quite small, but definitely they are not as miniature as in Lentibulariaceae. The genomes of carnivorous Caryophyllales were not smaller than their non-carnivorous relatives. Actually, they did not differ at all. But we found that the GC content in the family Droseraceae is adaptive, possibly because of the stabilizing effect of the GC content on DNA. Well, now we see that in this group, the evolution of genome size is probably not driven by nutrient limitation. And to decide whether carnivory is really associated with genome size decrease, we had to explore other carnivorous clades. So we made a study where we directly addressed this hypothesis and compared carnivorous plants in as many clades as possible. You have already seen the results in carnivorous Caryophyllales and results of Lentibulariaceae. In Lamiales, we have added Biblidaceae. Then we have added carnivorous plants in Ericales, Oxalidales, and Poales. As you can see, there is no clear trend, and truly, the carnivorous plants do not differ from their non-carnivorous relatives. The final conclusion for genome size and carnivory is therefore that despite some tendency for small genomes in carnivorous plants, carnivory does not seem to be connected with a general genome size decrease when we compare the carnivorous plants with their closest relatives. It might be that the carnivory is such a successful adaptation that it can actually counterbalance the nutrient deficiency and it can relax the pressure on the genome size. When we worked with the family Droseraceae, we knew about another interesting thing, because in some species of the Droseraceae, holokinetic chromosomes have been previously reported. The holokinetic chromosomes, unlike monocentrics, which has a localized centromere, which is a place for kinetochore formation, and microtubules, place for attaching of microtubules, holokinetic chromosomes form kinetochore alongside the whole length of the chromosomes. When the chromosomes are damaged, like by some clastogen, in monocentrics, the fragment is lost, while in holokinetic chromosomes, even small fragments are inherited and are kept in the plant or animal. In Droseraceae, the situation was quite confusing. There have been reports of monocentric and holokinetic species of Drosera. The chromosomal structure of Aldrovanda was completely unknown, and Dionea was supposed to be monocentric. We were quite sure that Drosophilum lusitanicum is monocentric. To analyze this, we used the flow cytometric method of detection of holokinetism. This method is based on counting diploid and tetraploid cells which are present in plants. The tetraploid cells are usually a result of currently dividing cells in the tissue, and their number is quite low in both monocentric and holokinetic species. When we apply clastogen like gamma irradiation, the situation changes. In monocentric species, the plants could lose broken fragments of chromosomes, so they stop the cell cycle in G2 phase, and the tetraploid cells are more numerous in the tissue. Therefore, the ratio between diploid and tetraploid cells increases. In holokinetics, the difference is negligible, or there is no difference at all. As I have said, we have been curious what the actual distribution of holokinetic chromosomes in the Masarykova, and we have also asked whether the genomes and chromosomes of the Masarykova exhibit similar patterns that were reported in other holokinetic groups, such as negative correlation between the genome sizes and chromosome numbers. As already mentioned, the Drosophilum lusitanicum was detected as monocentric. It can be confirmed on the direct observation because this species has very large chromosomes and the primary constriction is quite clearly visible. On the other hand, both species of Drosera were detected to be holokinetic, as well as Aldrovanda vesiculosa and even Dionea muscipula. As a result, we can say that holokinetism is an apomorphy of the whole family Droseraceae. When we tested or observed the parameters of the genome sizes against chromosome numbers, we have detected a negative correlation in one of the major clades of Droseraceae, or particularly the genus Drosera. This negative correlation is probably, or very probably, a result of holokinetic drive, which is resulting from competition for survival between chromosomal homologs in asymmetric female meiosis. Well, to conclude this direction of our research, we can say that holokinetism represents an apomorphy of the whole family Droseraceae, and that the holokinetic drive is probably active in one clade of the genus Drosera. My takeaway message for you is that carnivory does not include genome miniaturization automatically. The genome miniaturization in the family Lentibulariaceae is probably quite unique and intrinsic process, possibly the mutation of the COX enzyme, but this can change, and that the whole family Droseraceae has holokinetic chromosomes, and that the GC content is adaptive in response to environmental conditions, especially temperature annual fluctuations in the family Droseraceae. So, there are many people to whom I should thank, and I thank them, and I also thank you all for your attention.